Hello students, before C1 component, I will briefly go through what all the aspects we have discussed under recombinant DNA technology. We have discussed about the vectors and different forms of vectors, and also we discussed in detail about restriction enzymes, how they are useful in generating the DNA of our choice. So among the vectors, we have discussed in detail about how vectors are designed from plasmid. We also discussed about how PBR322 has been constructed, what are the major steps involved in the construction of PBR322, and we also appreciated how versatile this plasmid is by expressing the hormone somatostatin. Somatostatin is a simple hormone, very small in size. So the scientists have synthesized the DNA of somatostatin in the laboratory. After the synthesis, they have also introduced the restriction site at the end so that it will be useful for cloning in PBR322, and successfully they have expressed the hormone somatostatin. So that was a marvelous experimental work regarding the recombinant DNA technology to appreciate how best this technology is going to be useful. So the plasmid vectors for sure has some of its own limitations. To overcome the limitations of plasmids, we also have other vectors like Phage vectors, shuttle vectors, plant vectors required for plant biotechnology, animal vectors useful for animal biotechnology, yeast vectors, and finally we discuss about yeast artificial chromosome. So in the current video, we discuss about the Phage vectors. So now I have compared the plasmid vector as well as a lambda phage. I told you, so now this is the plasmid of PBR322. So this has an origin of replication. There is an ampicillin resistant gene. There is a tetracycline resistant gene. So now whatever the DNA we have insert, so we have to do the transfection with a host cell. Now the transfection has got a limitation. Suppose if the clone is bigger than 10 kilobase, then efficiency of transfection in the host cell decreases. That clearly put us in a negative side that we can only insert to this plasmid vector as big as 6 kilobases. Suppose if you want to have a DNA segment more than 6 kilobases, then the plasmid vectors are not the suitable one. So for us, if you want to understand the other genetic aspects of other genes which are really large, then insertion of such large DNA into plasmid vectors has got its own limitation. To overcome that, so we also have designed a lot of vectors from the Phage. What are exactly our phages? So now we do have discussed in detail about the lambda phage. Now the lambda phage has got a hexagonal head and then the tail, and there is a region which specifically binds to the host system. Now these phages are nothing but virus particle but infect bacteria. So the viruses that infect bacteria are called as bacteriophage, or the phages in fact. Now we do have several similar phages like lambda phage. They are the Psi80, Psi82, Psi21, Psi434. We also have a P2 and P229. These are all closely related phages which infect the host E. coli more or less in a similar fashion. So we are not interested in these phages because most extensive study has been done with lambda phage. Suppose if you try to understand the lambda phage, so usually this encodes a nucleic acid that is nothing but a double helical linear DNA structure. This particular one is encapsulated by a head protein, then followed by a tail region in fact. We will just study into the nucleic acid. Now I told you this nucleic acid is a linear double-stranded DNA structure. Now the complete DNA sequence is known. That is the advantage of this Phage vector, especially the lambda phage. It consists of 48,502 bases and has 40 genes within it, in fact. Now, how exactly? Now, you already know that in case of a plasmid, smaller the plasmid size, the advantage is so that we can insert a longer DNA sequences. To begin with, the Phage vector itself has got a very huge DNA structure. You will be wondering how such a DNA vector is going to be useful to incorporate still a larger DNA. So in order to understand that, we have to study the lytic cycle and a lysogenic cycle. This is the life cycle of lambda phage, and how this lambda phage will have its lytic cycle or a lysogeny. We will also find out some similarities between plasmid and a phage. Plasmid is a circular DNA but do not contain any other head-like structures. They also will get into the host system, and this particular plasmid, if it is in a host system, is advantageous to the host. So it will carry some additional phenotypic character to the host. In a similar way, lambda phage will also enter into the host system for its multiplication. So here it is not an advantageous. What exactly it does? In case of a lytic cycle, using the host machinery, it will multiply. At the end, it will lyse the host cell. So now, independently, plasmid by itself and the phage by itself cannot replicate. Both of them require the host system. Plasmids are beneficial. On the other hand, phages are, if it enters into the lytic cycle, then it will lyse the whole cell. Now I will first try to understand what are the lytic cycle and what is the lysogeny cycle. So before that, how this particular lambda phage will infect E. coli, that is the host. So it will not bind randomly to the E. coli host. It will bind specifically to a receptor. Now this particular receptor is from the gene LAMB. So now what LAMB gene does is that it expresses a transmembrane protein. Now this transmembrane protein is essential for the transportation of maltose to the cell. So it is a maltose transporter. So now this receptor is very important. So now the lambda phage recognizes this particular receptor and binds specifically to this particular region. Then it lies this membrane in this and introduce the linear DNA into the cytoplasm. Now the linear DNA is not the ideal one because this particular host has got an enzyme called exonuclease. So the exonuclease will cleave the terminal region. In fact, it will destroy. So in order to escape from the exonuclease activity, what happens is that even though it is a linear, at the end you are going to find 12 bases protrusion like an overhang. Now these 12 base pairs are highly compatible so that they come together and then they will get ligated. Now this particular region which these two things are ligated, that will result in the formation of a site called as a cos site, in fact. So we call this as a cos site. Now you will be wondering why the host chromosome is also circular in nature. It's a circular DNA because it has to escape from the host exonuclease. Similarly, even the linear lambda DNA also has to undergo circularization, otherwise it will be susceptible for exonuclease cleavage. So we will discuss about the role of exonuclease and its regulation in the later class. So now because of that, if you want to do a infection experiment with E. coli and lambda phage, better we have the medium rich in maltose. The more the maltose, so we are going to have more number of LAMB genes are going to be expressed in the membrane. And the optimum temperature for the infection is if you do the incubation at 37 degrees centigrade. And once it enters into the cell, now the question is, it has got two cycles. One is the lytic cycle and lysogeny cycle. So in case of a lysogeny, what happens is that this particular DNA will integrate into the host chromosomal DNA so that the cell survives. It is not undergoing any lysis. So once this cell undergoes duplication, So the chromosomal DNA also undergoes duplication. Along with the chromosomal DNA, even the viral DNA which got inserted will also multiply. Now that particular process is called as a lysogeny cycle. Whereas in case of a lytic cycle, what happens is that now this particular DNA undergoes replication, then it will also produce transcription and also lot more proteins required for the head region and also for the tail regions. Then there will be an assembly. So as a result, so in the lytic cycle what happens, the phage particle will multiply into 100,000s. Once it occupies the entire region, then finally it will lyse the plasma membrane and all the phage particles do come out. So now what happens is that unlike plasmid, so here it requires host for its multiplication. If it enters into the lytic cycle, that finally results in the lysis of the host cell. So now that is the huge difference between plasmid as well as the phage DNA. Now with this phage DNA, we will try to understand how this linear DNA is useful in developing a vector. I think I will close at this particular point.